Now we are discussing about sewage composition and chemical oxygen demand. Now coming to the sewage composition. The composition of sewage mainly depends upon per capita consumption of water and varies from place to place and season to season. The sewage composition can be studied under following two heads. One is chemical composition, second one is microbial composition. Now coming to the chemical composition. Chemically, the sewage consisting of approximately 99% of water and 1% inorganic and organic matter is suspended and soluble forms. Here we are observing lignocellulose, cellulose, proteins, fats and various inorganic particulate matter exist in suspended state, whereas sugars, fatty acids, alcohols, amino acids and inorganic ions constitute the soluble form. Here we are observe the soluble form and suspended form. Here lignocellulose, cellulose, these are the suspended state, whereas sugars, fatty acids, amino acids, these are in the soluble form. However, on an average, the sewage of town in our country contains about 350 ppm biodegradable organic matter, 52 ppm N2, 45 ppm potassium, 16 ppm phosphorus, salts of several heavy metals such as zinc, chromium, nickel, lead, etc. are also present above permissible levels in sewage. Next, coming to the microbial composition. The microbial population per milliliter of sewage may vary from a few lakhs to several millions. Various types of microorganisms, that is microfungi, bacteria and protozoa, collectively called sewage fungus, are known to grow profusely in sewage. In addition, viruses and many microalgal genera have also been recorded from sewage. Bacteria occurring in sewage are mainly intestinal and soil inhabiting, and their common types are coliform and streptococci, clostridia, micrococci, proteus, pseudomonas and lactobacillus. Here we are observing bacteria, viruses are phases nothing but, and fungi, protozoans are observed in the sewage water. Next, coming to the chemical oxygen demand, COD. COD is an empiric laboratory assay which measured the amount of organic matter contained in a water sample with the help of chemical oxidizing agents such as potassium dichromate and potassium permanganate. COD is a means of measuring of all organic matter that is biodegradable and non-biodegradable. This test is useful in studying pollution strength of domestic and wastewater. COD values are greater than BOD values. The open reflex method is suitable for a wide range of ways is discussed here. The main principle is the organic matter gets oxidized completely by potassium dichromate and silver sulfate crystal catalyst in the presence of sulfuric acid to produce carbon dioxide and water. The excess potassium dichromate remains after the reaction is titrated with iron molecule ferrous ammonium sulfate. The dichromate consumed gives the oxygen required for the oxidation of the organic matter. Here we are observing the carbohydrate material, proton dichromate gives rise to chromate molecule plus carbon dioxide and water molecule. What are the reagents required for estimation of COD? Here we are observing standard potassium dichromate. It consisting of 0.25 normality. Next one, sulfuric acid reagent. Third one standard ferrous ammonium sulfate 0.1 normal. Ferroin indicator, mercuric sulfate, potassium hydrogen phosphate, that is KHP standard. Now observing the procedure. First one here, first, second, third, fourth, fifth, sixth, seventh, eighth and ninth steps are observed. In the first step, take 2.5 ml water sample in tube and 2.5 ml of distilled water in another tube. Here we are taking two tubes. One tube is filled with sample and another tube is filled with distilled water. Takes 2.5 ml of distilled water and another tube take 2.5 ml of water sample. Next, coming to the second point, add 1.5 ml of potassium dichromate to both the test tubes, that is control as well as sample. Next, third one, carefully add 3.5 ml of sulfuric acid reagent to both tubes, that is sample and control. Next, third one, we are observing tightly close the tubes kept in COD digester at 150 degrees centigrade for 2 hours. Here we are observing COD digester. These tubes are kept in COD digester at 150 degrees centigrade for 2 hours. Then after cooling to room temperature, transfer the content to the conical flask. Here we are observing the material. This material is transferred into the conical flask. Then fill the burette with freshly prepared ferrous ammonium sulfate. Next one, add 2 drops of ferroin indicator. Here we are observing the ferroin indicator. Titrate the contents against ferrous ammonium sulfate. Continue the titration till the color changes to reddish brown. Here observing different colors, first yellow color, then change to green color, then change to red color. Continue the titration till the color changes to reddish brown, then calculate the COD concentration. Then after completion of experiment, calculate the how much amount of COD present in the given sample by using calculations. COD, here we are observing the formula, that is nothing but the COD mg per liter is equal to B minus A into N into 1000 divided by ml sample, whereas A is equal to volume of ferrous ammonium sulfate used for sample, B is the volume of ferrous ammonium sulfate used to blank, N is normality of ferrous ammonium sulfate. Next, normal levels. COD levels 0 to 5 mg per liter indicates very clean stream. Chemical oxygen demand 5 days at minus 20 degrees centigrade maximum, that is the parameters of COD. Inland surface waters 250 mg per liter maximum, and marine coastal areas 250 mg per liter.